A-Level Biology 9700, Paper 5, Variant 2, February March 2026. The third and last video on this paper, question number 3. And question number 3 is the Arman Bihu is a slow-flowing river that forms an aquatic ecosystem in Texas, North America. Some biologists decided to study two invasive alien species of aquatic plants that are commonly found in the Arman Bihu, water hyacinth, Ecomia gracefs, and Melanca. So, Althimanthera phylloxeroides, water hyacinth, and Melanca reduce biodiversity in the Arman Bihu. Water hyacinth is a floating plant that grows on the surface of the river. Water currents and the wind can move water hyacinth plants from one place to another. So Melanca has roots that attach the plant to the sea soil at the edge of the river. Melanca grows and reproduce to form dense mats on the river surface, which can trap floating water hyacinth plants. So show figure shows these plants. You can see 3.1 and 3.2 right over here. So the biologist carried out a survey at 69 locations in the Arman Bihu. At each location, both invasive alien species were present. So the biologist estimated percentage cover of water hyacinth and the percentage cover of Melanca at each of the 69 locations for these two variables. The biologist calculated uh, Pearson's linear correlation coefficient R as plus 0.389, which was significant as P less than 0.001. So complete the sketch graph in figure 3.3 to show the relationship between these two variables. Include x-axis labels in your answers. So here I'm going to, first of all, as we have to find out this linear correlation, so at x-axis, we will label the percentage cover of water and percentage water hyacinth. So it will be written on y-axis, x-axis, and on y-axis we will write as percentage cover of Melanca. So now we have to draw the a straight line, which will be, you can say, sloped line, which will extend from bottom left to the top right, because it will show a positive correlation. R is equal to 0.389. So it will be in this way. So there will be the straight line in this way. So it will show the positive correlation. R is equal to 0.389. So you have to label x-axis, y-axis, and then this line will show positive correlation. Now the next part of this question is the biologist carried out an investigation at 12 of the 69 locations in the Arman Bihu where both invasive alien species are present. The biologists wanted to test the hypothesis that the removal of Melanca from the Arman Bihu will reduce the abundance of water hyacinth. So shows a map of the Arman Bihu and the surrounding area. You can closely observe this figure 3.4, and you can see the figure 3.5 is there. At each of the 12 locations, a 3 m meter into 5 meter study area was marked out as shown in figure 3.5. You can see the area which has been marked over here, so you can see this one. So describe the method that the biologist used to select 12 locations at random from the 69 locations used in 3. So here what will happen, that is we will randomly select. So for this randomly selection, we will assign number 1 to 69 to all original locations and generate 12 distinct random numbers by using a random number generator. So I am going to write this answer as assign 1 to 69. 1 to 69 to all original locations and generate 12 distinct random numbers. Using here using a random number generator. So in this way, it will be done. Now the next is the 3 meter into 5 meter study area at the 12 locations was divided into 3 groups of 4. Each group of study area was treated in a different way as shown in the table 3.1. So treatment was number 1, removal of melanka and water hessian, and the details of treatment was there. And then removal of melanka, and then there was a control. So they have given the details of treatment with 3M into 5M study areas. At removal of melanka and water hyacinth at the start of the investigation, the biologist removed all the melanka and at the water, other plant species were not removed. The removal of melanka was repeated every week for 7 weeks. And then number 2, removal of melanka, only the start of the investigation, they removed all the melanka, water hyacinth, other plants were not removed. So the removal of melanka was repeated every week for 7 weeks. So the control was melanka, water, and other plant species were not removed. Nothing was removed in the control. So after 8 weeks, the biologist recorded the area covered by water hessian within the central strip of each study area as shown in figure 3.5. The area covered by water hessian was used as a measure of abundance. Now the figure 3.6 shows the results of the investigation. Error bars show 95% confidence interval, 95% CL. So here you can see the treatment 1 in which the main area covered by water hyacinth, it was it was 2 in treatment 1, but it was 1.55 or 1 around 1.6 was in the treatment 2, and in the treatment 3 it was 3.20 or 3 point something else. So it was about that. So these are the 3 different treatments in which they have shown this. The mean area covered by water hyacinth in location given treatment 2 is smaller than the mean area covered by water hyacinth in location given treatment 3. So use the data in figure 3.6 to calculate the difference as a percentage of the results for treatment 3. So here, as I have told you, the area which was for treatment 2, uh, was covered by the plant is it is about treatment to the area covered is around 1.60 or something between 1.55 and 1.60, and in treatment 3 so the control it is around 3.20. So we are using these two values to calculate the percentage difference. So it will be in this way. First of all, the difference. It is 3.20 minus 1.60 is equal to 1.60 meter square. Now the percentage difference, it is 3.20 minus 1.60 divided by 3.20 multiplied by 100. So this is the formula to find out this percentage. So it is around, you can say the answer can be 49.2% or between 49 to 50% can be the correct answer. So it can be acceptable. So if it is between these two values. So the next question is state which statistical test could be used to establish the statistical significance of this difference between locations given treatment 2 and the locations given treatment 3. So here which test in biology we use, that is the t-test. So here I'm going to write the student's t-test. So this is the answer, t-test. Now explanation for this is because you know that the t-test is used to compare the means of two independent categorical groups with continuous data. So I'm going to write the same as I have told you. Used to compare the means of two independent categorical groups with continuous data. Now, moving towards the next part of this question, which is part 4. The risk assessment for this investigation stated that the biologist should wear gloves to prevent an allergic reaction to the different plant species. So it's just one hazard, other hazard of the investigation, the risk associated with the hazard and the precaution that would you need to take. So here, I can see there are so many different risk factors over here. So the hazards can be the deep water, the river currents, and slippery banks. So all they can be the hazards. So I can write any one of them. And first of all, the hazards. So there is deep water over there. You can write this one, or you can write as the water currents or the river currents. And you can also write slippery banks. So it happens. So all these are the different hazards. Now the risk. What is risk with from these hazards? That is, uh, they can fall into the river or they can drown into the river. So I will write both of these. Falling into the river or drowning. So what should be the precautions? For this, there should be the precautions. Wear a life jacket. It will help not to drown. And secondly, uh, keep away from the steep edges or work in pairs. So it will be helpful that people will be there who can help to at any moment or in case of any mishap. Now the last part of this question is a conservation volunteer wanted to reduce the abundance of invasive alien plant species found in the almond Bayou. So the conservation volunteer analyzed data from the investigation and concluded that removing Malanka would reduce the abundance of water hyacinth in almond Bayou. With reference to figure 3.6 and other information provided about the investigation, evaluate the extent to which this conclusion can be supported. Well, uh, there can be the some you can see sporting arguments which can support this conclusion. So here I would go with the the in treatment one. You know that we have removed both plants were initially they still grew back to 1.9, which is higher than treatment 2. So the sample size is small, only 4 study areas per treatment group. So the 8 weeks is a short duration to assess long-term ecological dynamics. The fourth one, extraneous environmental variables, water flow, light, nutrient runoff, or seasonal changes were not controlled. So they should be controlled over there. So I would like to write in treatment one, Hessian, water Hessian, still grow, grew back to 1.9 meter scale, which is higher than second treatment. And the second point for here was the sample size is small. That was there were only 4 study areas for treatment. 4 study areas. Then the third 8 weeks is a short division to assess long-term ecological dynamics. Number 4, extraneous environmental variables were not controlled. As you know, water flow, light, nutrient runoff, or seasonal changes all day. So, well, student, it was all about this paper 5.2. Please like, share, and subscribe my channel to watch more videos on the May June 2026 papers. Thanks for watching.